Hi friends, today we discuss about ELISA. ELISA means enzyme-linked immunosorbent assay. It's mainly used to detect antigen present in the samples. Now various ELISA kits are available in our market. An example, HIV ELISA kits. So first of all we discuss about the basic principles of ELISA. So in the case of ELISA test, we first taking an antigen containing sample, that means we tested sample. So the tested sample containing some antigens. So this antigen is react with an enzyme linked antibody. So the antibody is linked with a enzyme. So the enzyme linked antibody and the antigen present in the sample is reacted and to form an antigen antibody complex. This is the first principles of ELISA. Then after the antigen antibody complex formed, to this complex we adding a specific substrate. The specific substrate is added to this antigen antibody complex. So the substrate is react to the enzyme. That means the enzyme is linked with the antibody. So the enzyme is react with the added substrate. So it's form an enzyme and substrate complex. Finally it's converted to a product. This product imparts some colors to the samples. So after the formation of enzyme substrate complex, it leads some colors. Mainly it's form a blue color to the samples. So this color is used to detecting the concentration or the presence of antigen in samples. So we using colorimetric method to detect the antigen's presence in specific or at specific wavelength. So next is methodology of ELISA. ELISA test we carried on a polystyrene microtiter plates. So the plate is contain 96 wells. So the figure is representing a polystyrene microtiter plates which containing almost 96 wells. So each wells we adding samples. So this polystyrene microtiter plate is used for ELISA test. So the first step in ELISA test is adding the antigen containing sample to the microtiter plates. So we adding first to the sample. So if we considering this is a single well of the microtiter plates, so we adding a antigen containing sample to the well. So it's contain antigens. This blue dots representing the antigens. So the antigens are most the antigens have more ability to attach with this microtiter plates. So the antigens are tightly bounded with the microtiter plates. Then second step is washing out of this unbounded antigen. So the unbounded antigens are always present in these wells. So the unbounded antigens are washed out by using specific buffers. So we commonly use the buffers are triphosphate buffer which have pH 7.4 and a detergent. So the detergent commonly used is TN20. The concentration of TN20 is varying. It's mainly from 0.01 to 0.05%. So the unbounded antigens are washed out. So the microtiter plates only contain the bounded antigens. So to this antigens we adding an enzyme linked antibody. So this black color is representing a enzyme linked antibody. So it's a enzyme linked antibody. So we represent antibody as a Y shape. So it's a enzyme linked antibody. So we add it into the microtiter plate. So it's react with the antigens present in the microtiter plate. So it's form antigen antibody complex. So enzyme linked antibody is added into this microtiter plate. So this in which it tightly bound with the antigen. So the antigen antibody complex is formed. The microtiter plate is always contain unbounded antibodies. So these unbounded antibodies are washed out by using some buffer solutions. So next step is washing of unbounded antibodies. So then finally the microtiter plates contain only the antigen antibody complexes. So to this complex we adding specific substrate. So next step is adding of specific substrate. This specific substrate is react with the enzymes. So we adding some specific substrate. So the red color is representing some substrate. It's a substrate, specific substrate. This specific substrate is added. So within the microtiter plate, so it's consider it as a microtiter plate well. So it has an antigens, bounded antigens. These antigens are always interact with the enzyme-linked antibodies. Enzyme-linked antibodies. This after the formation of antigen antibody reactions, we adding a specific substrate. So this specific substrate is react with the enzyme. So it's form an enzyme substrate complex. So it's form an enzyme substrate complex. So finally it's converted to a product. So we got the So the enzyme substrate complex is converted to a product. This product is impart a blue color to the sample. So after the formation of enzyme substrate complex, this reaction leads to the formation of products. These products leads a bluish color to the sample solutions. So next step, we adding sulfuric acid, that is H2SO4 to the microtiter plate. So the sulfuric acid convert the blue color to yellow color. So after the formation of yellow color, we analyzed the sample with colorimeter at specific wavelength. This is the basic method of ELISA. Now next, the enzymes which used in ELISA techniques. There are mainly two enzymes are commonly used. First one is HRP type and second one is AP type. HRP means it's a horseradish peroxidase enzymes. Then second one AP means it's a alkaline phosphase enzymes. Then based on these enzymes, there are two substrates are commonly used. First one is OPD. OPD means it's a O-phenylene diamine dihydrochloride, and second one is PNPP. It means P-nitrophenyl phosphate disodium salt. So the first OPD substrate is commonly bound with the HRP enzymes. So the enzyme HRP is commonly bound with the OPD substrates. It's form yellow color. This yellow color is determined with colorimeter in colorimeter at the 1492 nanometer wavelength. Then in this the enzymes alkaline phosphase is always bound with the PNPP substrates. It leads to the formation of yellow color. Then this is determined at colorimeter in at the wavelength of 405 nanometer. So it's the commonly used substrates and enzymes in ELISA techniques. Now next type of ELISA techniques. Mainly there are four types of ELISA techniques are present. The first one is direct method, then second one is indirect method, third one is sandwich method, and fourth one is competitive type method. So first type is direct method. In direct method, we using the microtiter plate. First we adding the antigen containing sample to the microtiter plate. Then to this antigen we adding antibody which is enzyme linked. That means the enzyme linked antibody is added to the antigen. Then it's form an antigen antibody complex. To this complex we adding specific substrate. So this substrate is always bound with the enzymes. Then this enzyme substrate complex formed. This enzyme substrate complex leads to the formation of color. Then it's analyzed to or analyzed with the help of colorimeter. This is the direct method. That means first we using antigen containing sample. To this sample we adding enzyme linked antibody. Then it's form antigen antibody complex. To this complex we adding a specific substrate. This substrate react with the enzymes and to form enzyme substrate complex. This complex leads to the colors of samples. Then this color is determined with the colorimeter. Next, second type, indirect method. In indirect method, we first using an antigen containing sample is first add into the microtiter plate. Then to this samples we adding a primary antibody. This primary antibody is not enzyme linked. So the first we adding the sample which containing antigen, then to this antigen we adding primary antibody. It is not enzyme linked, so the antigen antibody complex is formed. To this complex we adding a secondary antibody. This secondary antibody is enzyme linked. So first we adding the primary antibody. The primary antibody is first forming an antigen antibody complex. It's just like that. So it's forming antigen antibody complex. Then next we adding a secondary antibody. It is an enzyme linked antibody. So it's react with the primary antibody. So it's the secondary antibody. Say enzyme linked antibody. Okay, so the setup is first antigen, then we adding primary antibody. The primary antibody is not enzyme linked. Then next step we adding a secondary antibody. It is enzyme linked. So this enzyme linked antibody is attached with the primary antibody. Then finally we adding a specific substrate. This substrate is react with this enzyme and to form enzyme substrate complex, and it leads to the color of the sample, and this color is analyzed with colorimeter. This is the second type of ELISA. Now next type is sandwich type. In sandwich type we using antibody coated microtiter plate. To this antibody coated microtiter plates we adding the antigen containing sample. The dark spot is representing the antigen present in the samples. So the antibody antigen interaction is occur to form antigen antibody complex. So to this complex we adding an enzyme linked antibodies. So this enzyme linked antibody is always react with this antigens. So the entire setup is just like that. First is an antibody not linked to the enzyme. Then second is antigens. Then third is antibody. It is enzyme linked antibody. So the antigen is completely inner side. So the two sides are antibodies. So due to this setup, this technique is known as sandwich type. Antigen is middle, then two sides are covered with antibodies. So first antibody coated microtiter plate is used. To this microtiter plate we adding the antigen which contain samples. Then we adding an enzyme linked antibody. This enzyme linked antibody is interact with the antigens. Then we adding a specific substrate. This substrate is react with this enzyme and to form enzyme substrate complex. It leads to the color. Then we analyze it with the colorimeter. This is the third type of method, sandwich type. Okay, now next it's a competitive type. Fourth type is competitive types. So competitive types is commonly used when the sample containing less amount of antigens. So the presence of antigen is very less. We using competitive type of ELISA. So in this type we taking the antigen present in the sample and add to this specific antibody. So the amount of antigen is very less. Then we adding this specific antibody is interact with this antigen to form antigen antibody complex. Due to the less amount of antigen, there are many unbounded antibodies are present. So this entire mixture is Then we added into the microtiter plates. The microtiter plate is always coated with a specific antigens. So microtiter plate is coated with specific antigen. To this microtiter plates we adding the first mixture, that means the antigen and antibody mixture. So the unbounded antibodies are react with the antigen present in the bottom of microtiter plate. So the antigen antibody complexes are formed. Then we added a enzyme linked antibodies. Then finally we adding a substrate. This substrate is bind with these enzymes to form enzyme substrate complex. This substrate enzyme substrate complex leads to the colors, and this finally this solution or the sample is analyzed with the colorimeter. So this is the competitive type. So competitive type is mainly used when the sample containing less amount of antigens. Okay, these are the mainly about the ELISA.